In this lecture, we are going to see a study about enzyme inhibition kinetics. Yeah? So, in the previous lecture, we talked a lot about enzyme kinetics and some theoretical expressions that allows you to treat the experimental kinetic data when you are going to use enzyme as a catalyst. Yeah? So, in this one, we are going to deal something similar, but similar in the sense you are going to perform the same enzymatic reaction, but you are going to add some extra molecule into the reactor, so that will help you to stop the action of the enzyme. So I'm going to say what is an enzyme inhibitor. Yeah? So this lecture I'm going to show you a lot of theoretical expressions. So the best way to learn this topic is to assume these expressions are some mathematical rules that will allow you to explain the changes that you will observe in a reactor when you are going to deal with an enzyme. Yeah? So just to consider this as some mathematical expressions, and just assume the whole reaction is going to follow some set of rules, and the outcome of those experiments you can predict using these expressions which we are going to cover in this lecture. Yeah? So I'm going to give some definition for enzyme in what is an enzyme inhibitor. It's like, so they are nothing but enzyme inhibiting agents. Enzyme inhibiting agents, normally we add them purposely, so purposely added to decrease the rate of an enzyme catalyzed reaction. In the lecture 6 I already showed the job of an enzyme. Yeah? So what they do, it takes very large molecules, so they associate with large molecule and then they break them into smaller molecules. So these are products if you want, and then leaving the enzymes. So this is the job of an enzyme. So what's the job of an enzyme inhibitor? What's the job of an enzyme inhibitor? This is to, it inhibits the activity of an enzyme. So whatever the job performed by enzyme, if the enzyme is converting say like 10,000 or producing 10,000 molecules of product per unit time, if you add an inhibitor, it will decrease the speed so that the enzyme will start to produce maybe 10 molecules per unit time. So it's going to decrease the speed rate of the reaction or decrease the initial velocity. So that's the job of an enzyme. So the job of an enzyme is to diminish or to decrease the rate of the reaction, or in some cases to stop the action of enzyme. So or stop the job of the enzyme. Yeah? So that's the role of inhibitor, or if you want, you can call that the job of an inhibitor. Yeah. So they reduce the catalytic activity. So inhibitor will ultimately reduce this number. This is something I called as a turnover number. So once you have this inhibitor, it will decrease this number of an enzyme. So that's the job of inhibitor. And what is an inhibitor? You all know enzyme is a molecule, so you can express them in terms of moles. You know the molecular weight. The same rule applies for inhibitor. Inhibitor is a molecule, so which means you know the molecular weight. Obviously molecule have a nice molecular structure, and you all know enzyme have a beautiful structure. And I said to you enzymes are very specific in the lecture 6, and that's why I showed you some hypotheses. Imagine this is your enzyme, and So this part is your substrate. So they lock with each other. Yeah? And why that happens? Because enzymes have a definite molecular structure, and so do your substrate, which is another molecule. It has a molecular structure, and they complement each other so that they will lock to each other, forming this enzyme substrate complex. Inhibitor is a molecule. Say this is your enzyme, and this part will be your inhibitor. So inhibitor is something similar to your substrate. It has some particular shape, and they go and specifically bind with that enzyme. So again, you can apply that Fisher lock and key hypothesis. So eventually it will form some enzyme inhibitor complex. Yeah, so inhibitors are molecules. They have a definite molecular structure, and in many cases they have the structure of your substrate, so they can go and bind to an enzyme, and then it will create some enzyme inhibitor complex. So this is the basics you should know about enzyme inhibitors. So why do I talk about molecules and structures? I hope you all remember this Michaelis-Menten kinetics. Yeah? Enzyme substrate complex, it goes to enzyme plus products. So the rate of reaction, or the rate of catalytic reaction, depends on the concentration of the enzyme substrate complex, which is this one. Yeah. So if you manage to decrease the concentration of enzyme substrate complex, then naturally you are going to decrease the rate of the reaction. So if you add an inhibitor here, yeah, so, and if inhibitor and substrate have almost the same shape, say this is an enzyme, say this is your inhibitor, and this is your substrate. Yeah. Just assume some of your substrate associate with your enzyme. They form enzyme substrate complex, and then it goes to form enzyme plus products. But since you also have an inhibitor, this part, it will also associate with your enzyme. And since your inhibitor and your substrate, they both have similar structure, they can exactly go and lock into this particular space. Let me put some other color. This small space, yeah. So that can lock your impurity, so to form enzyme inhibitor complex. So since some of the enzyme are bound to inhibitor and to make enzyme inhibitor complex, so naturally the concentration of your enzyme substrate complex will decrease, so which means naturally the rate of reaction will decrease. So that's the whole concept of enzyme inhibitors. So why do we even care about enzyme inhibitors, or why do we even study this in this module? And that's because most of the drugs are enzyme inhibitors. So to complement this lecture, I'm going to upload the PowerPoint in Sulis, where I talked. I'm going to upload like maybe how many slides? Maybe 3 to 4 slides separately in a PowerPoint, where I talked about importance of enzyme inhibitors. I gave some examples. Okay, anyway, I can say it quickly. Like so, during the infection by HIV, like the lifetime of the virus, it depends on 3 enzymes. I'm not going to talk a lot about the biology part, but I'm just going to give, or just emphasize the importance of enzyme inhibitors, because I'm going to show some mathematical expressions explaining what is enzyme kinetics, where the inhibitors go and bind to the enzyme, and then I'm going to show some equations with some constant. So you have to know why do we even need to use those type of expressions. Yeah. So before going into mathematics, you should know the importance of studying enzyme inhibitors. Yeah. So for the lifetime of the HIV virus depends on these three enzymes. Yeah. So what these enzymes do, if you take this reverse transcriptase, so what it do is like it will copy the viral RNA genome, and then it will by combining free nucleotides, and then it will build a complementary DNA strand. So I have some nice animation, so I will upload the PowerPoint. Just go and check the PowerPoint. Yeah. And what this integrase will do, so once the reverse transcriptase it finishes its job, so it will oh sorry, this reverse transcriptase, what it will do is like it's going to copy the viral RNA genome by combining nucleotides, and then it will build a complementary strand. And once it finished building the complementary DNA strand, it's going to replace the original RNA strand with another DNA strand to build a DNA double helix. Yeah? If you don't want to go into biology, if you want to just think from engineering point of view, just think like the lifetime of this virus depends on three enzymes, and these three enzymes have to do some job. Yeah. And I'll just explain the biological part of the job of these enzymes. So, and what's the job of this integrase? So this particular enzyme will help to insert the viral DNA into our cell DNA. So this eventually causes the major cell destruction. So that's the job of the integrase. And when the third enzyme, HIV protease, what it do is like it's going to cut the polyproteins into functional parts. In fact, most of the virus replicates through this strategy by cleavage of the large polyproteins, and for that you need this particular enzyme HIV protease. Yeah. So how to kill the lifetime of the virus? So you can use some drug molecules that can stop the job of these enzymes. So drug molecules that can stop the job of these enzymes. Yeah. So you are adding some molecule purposely so that you are going to arrest the job of this enzyme, or you are going to stop the activity of these enzymes. Yeah. And there are few drugs like AZT and Paravir. Sorry for my pronunciation. So this one have a structure of nucleotide. So this reverse transcriptase, say this is your enzyme, say this is your nucleotide. So what's this one will take? So it will take your nucleotide here, and that's how it works, and then it build the complementary RNA strand. So this AZT, this molecule, this has the same structure of the nucleotide. So let's call this one as AZT, your drug molecule. So this one will go and lock into this enzyme, so which means it cannot accept or it cannot bind the nucleotides. So which means you stop the job of this enzyme, or you decrease the enzymatic action, yeah, by adding some inhibitor molecules that mimics the molecular structure of your substrate. In this case, it's nucleotide. Take the example of integrase. So integrase, imagine this is an enzyme, and this integrase, it has two magnesium ions here. So you need these two ions to integrate the viral DNA into human cells. So what we do is like we use a drug molecule, raltegravir. So it will go and bind to these two magnesium ions. So this is your drug molecule. So which means this enzyme cannot do the job. So it cannot integrate your DNA into the live cells. Yeah, viral DNA into the live cells. So raltegravir, this is a molecule, and what it do is like it inhibits the action of the integrase enzyme, HIV integrase. And then finally protease. Just assume this is HIV protease. So what it takes is like it, say this is your large protein. So this is your this enzyme. So that's your protein. So it will take your protein and then it cut them into two individual functional parts. Yeah. So what we do, we are going to use a drug. It's called sacubitril. So it has the same molecular structure of this particular part of your protein. So it will go and bind to this enzyme. So which means this enzyme cannot do the job. Yeah? So which means again you are using a drug to stop the activity of this enzyme, HIV protease. So this drug is called sacubitril. So whatever the drug molecules that can stop, which we used to stop the activity of enzyme, they are nothing but inhibitors. And it's very important to know how the inhibitors alter the enzyme kinetics. And there are few mathematical approaches to expose the actual mechanism, and that's what we are going to study in this lecture. Yeah, so this is a really an important topic, and again this is an engineering topic, so we will deal this topic purely from engineering point of view. We will have some mathematical expressions that will allow you to expose the actual mechanism, yeah, like where your inhibitors are binding either to enzyme or to some other location. So those things we are going to expose using a set of mathematical expressions. Yeah? And few more properties of enzyme inhibitors. So I said to you, you have an enzyme and then you have your inhibitor here. So this is enzyme inhibitor complex, and then you can separate them, yeah, into enzyme and inhibitor. So this type of process is called like reversible. So enzyme inhibitors, they can be reversible, and some cases it may be irreversible. Just imagine you have an enzyme, and once your inhibitor bind to this enzyme, it might alter the shape of the enzyme. So which means we call this as protein denaturation or enzyme denaturation. So once it loses the shape, it's very difficult to recover the shape of to retain the structure of the enzyme. So these are irreversible. So this type of inhibitors are called irreversible inhibitors. So this is some property of enzyme some property of inhibitors that you should know. You all know the properties of enzyme. I think I covered in detail in lecture 6 and also part 1 of lecture 7. Yeah. So we are going to type of how do you call different type of inhibition effect. I'm just checking my notes. I want to know whether I'm going correctly in the order. I already talked about agents, secondary, reversible, irreversible. Okay, yeah. So I said to you you have an enzyme and you have an inhibitor. So it can go and bind to this enzyme, forming an enzyme inhibitor complex. It's always not necessary that your inhibitor bind only to your enzyme. Yeah, imagine you have another case. You have an enzyme, you have a substrate. Say this one is your substrate, this is your enzyme. They combine to form enzyme substrate complex. Yes, and just imagine you have a inhibitor. Let's call this one as inhibitor, and they can bind exactly to this place, so it can form. So this is your inhibitor, this is your substrate, this is your enzyme. So it can form enzyme substrate inhibitor complex. So this one can also decrease the rate of your reaction, and this one can also decrease the rate of your enzymatic reaction. Yeah? So these are some way you can imagine how you can decrease the rate of an enzymatic reaction, and what's the job of your inhibitors. Yeah, so your inhibitor can bind to your enzyme, forming an enzyme inhibitor complex. That's one rule. Or your inhibitor can bind to enzyme substrate complex, and then it can form enzyme substrate inhibitor complex. That's another rule. So these are the two things you have to remember. And in some cases your enzyme can, I don't know how to draw that one, it's a bit complex. Say you have an enzyme like that. So let's call this one as enzyme. Let's say this one as your substrate. So they can combine. See, I'll try to draw. So this one is your enzyme substrate complex, and you say you have your inhibitor here. So it can bind to the enzyme here, So this is your inhibitor, this is your enzyme. So this is your enzyme inhibitor complex. Yes. And this is your enzyme substrate complex, this part. So just assume your inhibitor, which is here. So this is your inhibitor, this is your inhibitor. So it can bind to this enzyme substrate complex. So that's your inhibitor, that's your substrate, that's your enzyme. So it can form enzyme substrate inhibitor complex. So both will decrease your rate of your reaction. Yes, to say plus product, say this one is your product. So what it do is like your inhibitor will bind directly to the enzyme, making an enzyme inhibitor complex. So thereby it will decrease the action of your enzyme, which means the rate of reaction is going to decrease. And then your inhibitor can also bind to your enzyme substrate complex. This is your enzyme substrate complex. It can, and this is your impurity, it can bind to your enzyme substrate complex to form enzyme substrate inhibitor complex. Yeah? So this one can decrease your enzyme. So depending on the way inhibitor act on the enzyme or the overall reaction, you can classify this enzyme inhibition process into 3 different types. I'm going to write one by one, and I'm going to go a bit slow because this is a bit tough. Last year the students who do not have an engineering background, they really struggled to understand the concept. So I'm going to go really slow this topic. Yeah. So depending on the way, or if you want, you can call pattern, yeah, how the inhibitors decrease the rate of reaction. They classify the enzyme inhibition process into at least 3 different types. One is competitive inhibition, another one is uncompetitive inhibition, and another one is non-competitive inhibition. So we are going to study this complex topic by just making some nice cartoon images that will allow you to understand, yeah, how these things work. Yeah, like we try to draw bit clearly. So what is this competitive inhibition? So you see the word here, competitive. So just going to draw an enzyme for you. Let's call this is your enzyme, and say you have an inhibitor. Say this is your inhibitor molecule. Enzyme is a molecule. These are cartoon images. The real enzyme structure contains many atoms and it looks so complex. Yeah? Nobody can draw that using a paper and pen. So inhibitor is your molecule. So they will go and bind to this pocket. Yeah. So whenever you are talking about enzyme, just imagine everything is about pockets. Yeah, like scientifically we call them as van der Waals pockets. So this inhibitor will go and sit in this pocket, forming enzyme inhibitor complex. So this is your enzyme, this is your inhibitor. So the whole is enzyme inhibitor complex. Yes. And then the remaining enzymes will, just assume this is your substrate. So the remaining enzymes, so some enzymes will form enzyme substrate complex. The remaining enzymes will form, some enzymes will form the enzyme inhibitor complex. The remaining enzymes will form enzyme substrate complex. Yeah, and you all know this one will goes into enzyme plus products from the Michaelis-Menten kinetics. Yeah? So what you learn from this one is like you have an enzyme. You have one site here, which is here. Let me change the color. So this part of your enzyme, you can call them as an active space or a packet. Yeah. Let me do it a bit clearer. So this part you can call it as active space. So or a site. Yeah. So both your substrate and your inhibitor, they both compete for this one particular site. So that's why we call this as a competitive inhibition. Yeah. So try to check the notes. I have a better image in the PowerPoint slide. So also use the PowerPoint slide. So this is, in this lecture I'm just trying to explain the concept behind competitive inhibition, uncompetitive inhibition, and non-competitive inhibition. Yes. So but this, you have to understand the how do you call the actual physics of the process. Yeah. So it's not very easy. So so whatever the cartoon which I am drawing here, like you have a like active packet that will take both substrate, it can also fit your inhibitor complex. Everything happens at molecular level. Yeah. So that's why we are trying to understand the whole process using this simple diagram. So whenever you think about competitive inhibition, just assume you have an enzyme, which is a molecule that have a nice structure. It has some particular spot that can take both substrate and inhibitor. So which means it can form either a enzyme substrate complex or it can form enzyme inhibitor complex. And both of them are going to, so if once you have this enzyme inhibitor complex, which means you naturally are going to reduce the decrease the concentration of your enzyme substrate complex. So which means you are going to decrease the rate of the reaction, enzymatic reaction. So this type of inhibition kinetics, we call them as a competitive inhibition, or we can and then we will go on to uncompetitive inhibition. So uncompetitive inhibition, say this is your enzyme. Say this is your enzyme, and say this is your structure of your substrate, and say this is the structure of your inhibitor molecule, substrate molecule, enzyme molecule. So so what will happen is, like, you can see the active space here. Yeah, I'm just drawing some arrows to show you. So substrate can go and bind with enzyme. It can form enzyme substrate complex, and since I think this is a very bad diagram. Just assume this is your enzyme. So once it bind, your inhibitor can go only to this particular spot. So it can bind only to the enzyme substrate complex. So inhibitor will bind only to the enzyme substrate complex. So which means your inhibitor will not compete for the site that can fit your substrate molecule. So that's why we call them as uncompetitive inhibition. That's why we call them as uncompetitive inhibition. So finally we go to non-competitive inhibition. Non-competitive inhibition. So this is slightly complex one. So this one is like you have an enzyme and you have some space. Say this is your substrate. Say this one is your inhibitor. So your inhibitor, sorry, how to draw inhibitor like that, yeah. So your substrate can go with this complex, with the enzyme to make obviously this enzyme substrate complex. Yeah, and then your inhibitor say this is your inhibitor. So it can go and bind to this enzyme substrate complex. Forming enzyme substrate inhibitor complex. And then again you come back here, just assume your inhibitor goes to your enzyme directly here, and then it can also form enzyme inhibitor complex. Yes, and then your substrate can go and attack this enzyme substrate complex to form again this one, enzyme substrate complex. So this type of and naturally if you have an enzyme substrate complex here, it will go to enzyme plus products, but the rate of reaction will be very low because your enzyme is already attacked by your inhibitor, forming enzyme substrate complex by more than one route. Yeah, one is you form enzyme inhibitor complex, another is enzyme substrate inhibitor complex, and substrate can act on enzyme inhibitor complex, and then again you will form more enzyme inhibitor complex. So eventually you are going to decrease the rate of the enzymatic reaction. So this type of process we call them as non-competitive inhibition. So these are some experimental facts that really happen when you are going to deal with enzymatic reaction. Yeah? So your inhibitor can act on an enzyme and stop the activity of an enzyme in more than one possible way. At least, mm, in many number of ways. In this lecture, we are covering these three different types. Yeah, or three different route. So one is competitive inhibition, another is uncompetitive inhibition, another is non-competitive inhibition. Yeah. So when we when we are going to deal just with enzyme and substrate, we know your initial velocity is given by Michaelis-Menten expression. But when you are going to deal with a competitive inhibition, your rate of your initial velocity is given by, we don't have to derive this expression because it's time consuming, but you should know Km multiplied by alpha, where alpha is equal to 1 plus impurity concentration by Ki. So what is this Ki? So this is impurity concentration, which you know. This is a bit complex topic, yeah, but you have to learn. You should have some patience to learn this topic, yeah. So when you are going to deal with competitive inhibition, your initial velocity is given by this expression. So your substrate concentration is going to change with, your initial velocity is going to change with your substrate concentration in presence of a competitive inhibitor, and it's going to follow this mathematical expression. Yeah. So Km multiplied by alpha, where alpha is concentration of your impurity divided by Ki, and this Ki, how this one comes, I'm going to write the actual reaction, enzyme plus substrate. Enzyme plus products plus inhibitor enzyme inhibitor complex. So this Ki is an equilibrium constant. Is here, and that's how you write the reaction. Yeah? So this particular expression was derived based on this reaction. So enzyme plus substrate give rise to enzyme substrate complex, which goes into enzyme plus products, and then your enzyme is attacked or bind to an inhibitor, and then it can form an enzyme inhibitor complex. Yeah. So for this type of reaction, your initial velocity will be given by this one. So let's call equation A for competitive inhibition. You don't have to derive to study the, if you want I can derive, but it's really not necessary for this module, because it's very complex and it's very long one, and it won't serve the purpose, so for this module you have to know, like if I give the initial velocity at a different substrate concentration, and if I ask you to tell me like what type of inhibitors you are dealing with, is it a competitive inhibition or uncompetitive inhibition or non-competitive inhibition, you have to tell to me by solving the data. So for that we need some mathematical expression. So only I'm going to give that expression that will allow you to expose what type of inhibition mechanism is happening, like your inhibitors are attacking only enzyme, or your inhibitors are binding to both your enzyme and enzyme substrate complex, yeah, or your inhibitors are binding only to the enzyme substrate complex. So you have to figure it out. So I will give you the mathematical expression, and then I will show you how to how to mathematically solve or how to deal with the experimental data, yeah, and then identify what's the actual mechanism is happening, yeah? So, or what's really where your inhibitors are binding either to your enzyme or enzyme substrate complex or through some other way, yeah? So it's a bit tough topic, yeah, especially if you don't have a mathematical background. It's going to be even more tough, yeah? So if you're going to deal with uncompetitive inhibition, so I already said your enzyme plus your substrate, it gives enzyme substrate complex. It gives rise to enzyme plus products, and then your inhibitor, it acts only on your enzyme substrate complex, and then it forms enzyme substrate inhibitor complex. So your Ki is here, equilibrium constant corresponding to your impurity effects, yeah, that inhibitor. So Ki is is concentration of your reactants divided by concentration of your products. So this is, I think everybody knows this one, yeah, for equilibrium reaction. So that's how you write the constant, reaction constant. I hope you are all following this one. It's a bit tough, but once you solve the problem, then you will know, you will realize that this is not that difficult, yeah. So people already derived an expression that allows you to relate your initial velocity with your substrate concentration for this type of inhibition. So inhibitor that acts for this type of inhibitor, so which bind to enzyme substrate complex to decrease the enzymatic activity. The initial velocity is related to the substrate concentration by the relation alpha s plus km. Again, alpha is the same, 1 plus your impurity concentration divided by Ki. So let's call this as equation B. And finally, non-competitive inhibition. So what happens here? Your enzyme plus substrate, it goes to enzyme substrate complex. You get enzyme plus products. And then your inhibitor can act both on enzyme substrate complex to give enzyme substrate inhibitor complex, and then your inhibitor can also bind to enzyme to make enzyme inhibitor complex, and then your substrate can bind to enzyme inhibitor complex to give enzyme substrate inhibitor complex. So throughout this lecture notes I showed this arrow, which means they are reversible. So people used that assumption, this is reversible process, while deriving this expression, yeah. So for when you're going to deal with an inhibitor that can bind both to enzyme and also to your enzyme substrate complex, so in that case the initial velocity is given by alpha plus km. Again alpha, this is km is again your Michaelis Menten constant, yeah. So alpha is is equal to 1 divided by inhibitor concentration by Ki. So in this case, Ki, you can see inhibitor is acting both on enzyme substrate complex and also on enzyme inhibitor complex. So in this case, Ki will be equal to concentration of enzyme by concentration of inhibitor divided by the product concentration, which is also equal to enzyme substrate complex inhibitor concentration divided by product. So this part corresponds to this side of your reaction, and this one corresponds to this side of your reaction. So it's bit tough, but you will know how soon. You will know how to use this expression. So let's summarize this equation. Yeah. So let's say pure, which means no inhibitor. So that's given by Michaelis-Menten kinetics. When you have a competitive, when you experience competitive inhibition, then mu will be equal to mu max S Km. I think check, I don't want to do any mistake. Check my notes. So alpha will be with Km, yes, plus your substrate concentration. For uncompetitive inhibition, this alpha will go to your substrate. Mu is equal to mu max Km plus alpha multiplied by your substrate concentration. For non-competitive, you have to learn this. We are going to solve some problem in the exam. I might ask you some question, so like I might give some data between that correlates the initial velocity with the substrate concentration, and then I'm going to ask you which one is pure, which one is affected by some inhibitor, and what type of inhibitors you are going to deal with, whether it's a competitive inhibitor or a non-competitive inhibitor or a uncompetitive inhibitor. So you have to tell me. So these are the equations. I think so how to, just imagine I give you some data, say initial velocity, say substrate concentration, initial velocity of enzyme, another initial velocity. You have some number here. So say this is pure, this is with some inhibitor, say inhibitor 1, and this is in presence of inhibitor 2, and another mu value, say in presence of inhibitor 3. If I ask you to identify what type of inhibitor, say like whether inhibitor 1 is competitive or uncompetitive or non-competitive, you have to tell to me how like what type of inhibitor it is. So how to identify what type of inhibitor. So it's very simple. All you need is to go back to your Lineweaver-Burk plot. So let's take a competitive inhibition. Mu is equal to mu max, yes, divided by alpha Km plus substrate. You can linearize this into 1 by mu alpha km by mu max into 1 by substrate plus 1 by mu max. Yeah, so this is like y is equal to mx plus c. So this is your y value, and so this is your m value, and this is your intercept. Yeah. So for competitive inhibitor, your intercept will be equal to 1 by mu max, and your slope will be equal to alpha km by mu max. So so you can linearize the expression that corresponds to, that describes the how your initial velocity changes with your substrate concentration in presence of a competitive inhibitor. Yeah, and you can do the same for uncompetitive inhibitor. So what this inhibitor will do, yeah, I said to you, it will attack only the enzyme substrate complex, and in that case the initial velocity that is given by the relationship between initial velocity and substrate concentration. Yeah, so it's given by alpha into s plus km. So you can linearize them into 1 by mu is equal to km by mu max 1 by substrate concentration plus alpha by mu max. So this will be your y, so this part will be your m, so this will be your intercept c, so mx plus c. So your slope will be equal to km by mu max, and your intercept will be equal to alpha by mu max. I hope you all know how to use the graph sheet and plot your data. Yeah? So that's how we are going to deal with uncompetitive inhibitor. So, and what about non-competitive inhibitor? In fact, this enzyme inhibitors and your mass balance, these two are the toughest topic to learn and understand in this particular module. Yeah? And what about non-competitive inhibitor? So your non-competitive inhibitor can bind both to your enzyme and also to your enzyme substrate complex. Yeah? So in that case, your initial velocity will change with the substrate concentration, and it will follow the relation alpha km. So you can linearize this again like Lineweaver-Burk plot. All you need is to take the inverse and expand. Maybe for this one I can show how to do it. Mu max multiplied by x. So you can expand this one. This one is a bit more complex, yeah. So substrate get cancelled. So you will end up with one expression like mu max plus alpha km by mu max into 1 by your substrate concentration. So this is your y, this is your c intercept, so this is your slope, and this is your x value. So your if slope will be equal to alpha km by mu max, and your intercept will be equal to alpha by mu max. So that's how you are going to calculate these values, yeah, like So these are mathematical expressions. So but at the end of the day, if you look at this expression, say this one, you can see your y is 1 by u and your x is 1 by substrate concentration. This is for non-competitive inhibitor. Yeah? And if you go to uncompetitive inhibitor,